Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview authors from our feature articles, papers of the month, or other articles published in that month's print issue of the Diabetes Journal. We also have a Sweet Talk segment where we get a little creative in our scientist and topic interviewees. My name is Darleen Sandoval, and as always, I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are you all doing today?
Doing good. How about you, Kirk?
I'm good. How are you guys?
Should I let all of our listeners know that Kirk just completed his first marathon? What was that, 2 weeks ago now? Last weekend?
It was last weekend. I did the shuffle. Yeah.
Yeah. Nice. Boston qualifier?
Think so. Just under qualifying time. I don't know if I got the cutoff or not.
Yeah.
Nice.
Yeah.
What is the shuffle?
Super cool.
Oh, at the very end, it's less picking your feet up and more just shuffling them forward.
That's not the name of the race. That's not the one you competed in.
No.
Was it in Alabama, or where'd you go?
It was Gulf Shores, Alabama, which is that little nub down by the panhandle of Florida. So
very nice.
Yeah, cool. Well, speaking of sports, are y'all Olympics fans? Did you watch any of the games? What was your favorite event or sport to watch?
I'm a sucker for downhill skiing.
Yeah.
But I also love the speed skating, right? Because there's something about
so interesting.
Yeah. Yeah.
It's
also
so different and
yeah. What is it about the Winter Olympics and strapping knife-shaped things to your feet?
I didn't see it, but there was a clip of a lady that got clipped in the sunglasses or the protective eyewear that they're wearing.
Yeah, her sunglasses saved her. You're right. That was a short track speed skating, I think.
Yeah.
Yeah, that was
crazy.
Wow. Kevin, do you have a favorite?
Well, I'm just struck by what went around on social media. I didn't actually watch the race, but Johannes Klæbo, Norwegian, and his performance at the games, to where he's the first to win 6 medals.
Yeah.
And he did a lot of what I understand to be cross-country skiing events. And there's a video montage or GIF or something on social media that shows him sprinting uphill in skis at 11 miles an hour. And I was, that's just amazing. And his performance was just phenomenal. So
the Nordic events are really interesting because they're so, like, one athlete can do such a variety of events, right? So here he was at 6-minute mile going uphill in the skis, but he also won the 50K, which is a couple of hours of skiing.
Yeah, exactly. And I just can't imagine. I mean, this is coming from a guy that has trouble just standing on skis. So I can't imagine doing what he's doing. And then, of course, you can't leave out the U.S. Olympic hockey teams.
Yeah,
both the men and the women winning gold, and the women dominant performance in the last several years in that event, and then the men finally getting an Olympic gold since the Miracle on Ice in the 80s. So that was really cool to watch.
And almost to the date, right? To the day. Yeah, that was pretty cool.
Yeah. I was excited about it, even though and I think this happens a lot, but the event that ended up in the Olympics is not really like what is done widespread competition, but the Skimo events. Did you all watch any of these?
That was cool.
Yeah. It's pretty cool. And the Americans that did the mixed relay for the Skimo event are trail runners. And so I've been following Anna Gibson for quite a while. She qualified I think she made it to the semifinals of the 1,500 meters in the Olympic trials for the Summer Olympics. And then she's like a vertical. So there are these events in trail running called vertical kilometer runs. They're not necessarily a kilometer. They vary in distance, but it's essentially a race up a mountain. And she is like amazing at this. So it's really cool to see somebody with such varied talents, and it was really cool to see them do well.
I'm just a sucker for watching people do something really well.
Yeah.
Right. Even if I don't even understand how they do it or the motivation, but watching someone be at the very pinnacle, that's hard not to get geeked out about.
Yeah. And she's interesting because, and I think Cam is this way too, but I know less about him, but her relay partner, but she is one that is just like super stoked and excited and enthusiastic about competing, right? Like it's not about crushing your competition. It's really about just enjoying competition and enjoying the process of it all. So it's really cool. So our productions director makes a vote for snowboard cross, which is a good one. It is a good one. It's pretty crazy.
And you can throw some elbows, right?
Yeah. All right. Well, thanks, Wyatt, for that contribution. I guess we should talk some science. So, Kirk, tell us about our data publication interviews today.
Yeah, so we actually have 2 articles that focus on the islet. The first is Amino Acid Sensing by the alpha cell mitochondrial phosphoenolpyruvate cycle regulates intracellular calcium levels without affecting glucagon secretion. For that article, we were joined by senior author Matt Merrins from the Yale School of Medicine. And then in the second article, we discussed activating muscarinic receptor signaling in intrapancreatic neurons is required for parasympathetic cholinergic control of pancreatic cell function with first and co-corresponding author Noah Levi, who is a graduate student in the group of Dr. Alejandro Caicedo at the University of Miami Miller School of Medicine.
That was really cool because it's 2 very different perspectives of islet regulation, right?
It was. Yeah.
It was really fun. Kevin, tell us about our Sweet Talk segment today.
Yeah, we're previewing the upcoming American Diabetes Association scientific sessions. Is going to be held in June of this year in New Orleans. Each year, the scientific sessions, I'm sure Kirk and Darleen, you know this, they offer a first look at the studies, therapies, and scientific debates that will shape how we're all thinking about diabetes and metabolic disease. Joining us to walk us through what we can expect at this year's meeting is Dr. Mark Atkinson, who is serving as the chair of the Scientific Sessions Programming Committee. We'll cover what's different this year, what themes are emerging across the program, and why this year's meeting in New Orleans is one that you will not want to miss. So stick around and find out more.
Yeah, being in New Orleans in June is not always my favorite, but it's a really good conference center. It's a great city. So
Absolutely.
Yeah, it's gonna be, it'll be a good one.
Yep.
All right, well, let's dive in.
Today, we will be discussing a paper published in the March issue titled Amino Acid Sensing by the alpha cell mitochondrial phosphoenolpyruvate cycle regulates intracellular calcium levels without affecting glucagon secretion. We are joined by senior author Matt Merrins, who is the Joseph F. Hoffman Professor of Cellular and Molecular Physiology at Yale School of Medicine. Welcome, Matt, and thank you for sharing your work with us today.
Thanks for having me.
So, Matt, we like to start with an icebreaker question. So I came up with a new one today while I was walking the dog. That's it. That's when we get our most inspirational moments. If you could acquire any new skill in the world, it could be science related or personally, what would it be?
Well, I've always really wanted to learn how to swim well. And now that I live right by the ocean, it's across the street.
Yeah.
I'm realizing that my swimming skills are not where they need to be. My kids are doing really well, and my wife was a swimmer, but I could use some work. So I think that's the one that's top of mind.
Yeah, and it'll be useful for your
Although
new location.
This time of year, I don't know if you need those skills right away.
No, if we could just wait on that. That's a polar plunge. There's ice in the Long Island Sound right now.
Oh. Yeah. So, Matt, I'd like to give you the opportunity here to maybe talk about other collaborations or authors who helped you in generating this work.
The author I'd really like to highlight is Arlie Jin, who led the study. And he was a graduate student in my lab and had the really challenging task of coming into the lab right when COVID started. So the entirety of his PhD almost was done during COVID, but he ended up with a really great PhD, published 2 first author papers, one in eLife and this one in diabetes. And he's gone on to a terrific postdoc in Liangyi Chen's lab at Peking University.
All right. So, Matt, why don't you tell us, before we get too much into the science, why don't you tell us about the big gaps in knowledge that you wanted to address here?
Sure. So the big picture is that we know alpha cells are really important as amino acid sensors, but we don't understand how each amino acid regulates glucagon secretion. The reason we understand alpha cells are amino acid sensors is because, well, the liver catabolizes amino acids in response to glucagon. So I guess what I'm trying to say is alpha cells are amino acid sensors that release glucagon, and then glucagon signals to the liver to suppress the amino acid levels in the blood.
So it's like a regulatory circuit, right?
Yes, it's a regulatory circuit. But then we don't understand how the levels of the amino acids in the blood affect glucagon release.
So let's move on. And now I want you to talk about your other collaborators, but also to give us an overview of your research program and then kind of how this specific project grew out of it.
So in general terms, my lab is studying how nutrients stimulate hormone secretion from pancreatic islets. And we first got interested in studying amino acids and their effects on insulin release several years ago in a collaboration with John Campbell's lab. And at the time, the field knew that amino acids stimulated insulin release. But in our work with John, it became clear very quickly that the amino acids were not working directly on the beta cell. They were actually activating the neighboring alpha cell, and then paracrine signaling would stimulate insulin secretion, even in the fed state. And so our contribution to those studies, which we did with John's lab, was to show how specific amino acids work at the single cell level. And there are a few different amino acids that we were looking at that are relevant to this paper. So one of them, in the first paper we did, which was a collaboration with Megan Capozzi, then a postdoc in John's lab, and she now has her own lab. Megan found that alanine and arginine were really important for stimulating glucagon release from alpha cells. And the new part was that if she either genetically or pharmacologically blocked the beta cell GLP-1 receptor and the glucagon receptor, she would block the effects of these amino acids on insulin release. And we could also show that blocked their effects on cyclic AMP signaling, which is the mechanism of that effect. So that led us to a second paper, which was done with Kim Al, also in John's lab, where we actually looked at what was going on in the alpha cell itself. And we found that arginine and alanine were capable of raising calcium in alpha cells, and that could synergize with GIP, which is a gut incretin that binds to a receptor on the alpha cell surface. And the incretin raises the cyclic AMP, the amino acids raise the calcium, and then those two synergistically stimulate glucagon release. And then finally, I have to introduce one more amino acid, which is relevant to this story, and that's leucine. So leucine is special because it's the only amino acid that can stimulate insulin release at low glucose. And the way it works is that leucine's an activator of glutamate dehydrogenase, which is a mitochondrial enzyme that fills the TCA cycle with carbons from glutamine and glutamate. And the classic explanation of how leucine works on beta cells is that it stimulates mitochondrial ATP production to close the ATP-sensitive potassium channels, which depolarizes the plasma membrane of a beta cell and stimulates insulin release. But what Hannah Foster in my lab found was this is not how leucine works at all. And the clue that we had is that leucine's very good at stimulating anaplerosis, which is just a fancy term that means the net filling of the TCA cycle with carbon. So those carbons that are coming from glutamine going to glutamate and then into the TCA cycle through glutamate dehydrogenase, fill the TCA cycle. And that anaplerosis is serving a really important function that's not mitochondrial ATP production. So everyone's familiar with mitochondria as the powerhouse of the cell for their ability to make ATP, but mitochondria also have to produce products and signals that they send back out into the cytosol. And so one of those signals that we thought could come from leucine stimulation of glutamate dehydrogenase has to do with the mitochondrial producing a molecule called phosphoenolpyruvate, or PEP. And there's an enzyme, the gene name is PCK2. It's called mitochondrial PEP carboxykinase. And what it does is it can use the power of anaplerosis to pull from the oxaloacetate pool and convert it to PEP, and then kick the PEP back out to the cytosol. That mitochondrial PCK2 enzyme was shown in 2009 by Dick Kibbe's lab to be important for insulin release. And so what Hannah did is she knocked out that enzyme specifically in beta cells and found that leucine could no longer stimulate insulin secretion. She also knocked out one of the isoforms of pyruvate kinase, PKM1, and even at low glucose, she found that leucine could no longer affect insulin secretion that way. And so we started to form a model in the beta cell where the way that leucine works is it's filling the TCA cycle with carbons, forcing the mitochondria to make phosphoenolpyruvate, which serves as a substrate for the pyruvate kinase reaction. And that we found was locally controlling KATP channels in the plasma membrane. And so that's how we think leucine actually works. So a question we had leading into this paper is does leucine work the same way in the alpha cell that it does in the beta cell? Does mitochondrial PEP control alpha cell pyruvate kinase and alpha cell KATP channels? And then how does leucine interact with alanine and arginine? So that's what got us to the place that we're at now.
So then tell us about your results. So you initially did some perfusion experiments looking at the effect of the 4 different amino acids. The first focus was calcium flux. So tell us a little bit about those methods and what you saw.
So going back to this collaboration with John's lab, we had built tools to study alpha cell function, looking specifically at calcium and cyclic AMP. And so it was easy for us to repurpose those for this paper. And what we did is we looked at the effects of alanine and arginine, and we showed that they stimulate alpha cell calcium influx quite strongly, and they do so at their physiologic concentrations. And when we did a perfusion assay, where we looked at hormone secretion from the islets, what we found is that there was a corresponding stimulation of glucagon release. Leucine has an entirely different effect, though. It's inhibitory to the alpha cell. And so when leucine is applied with alanine and arginine, leucine can inhibit calcium influx stimulated by alanine and arginine. And leucine can also inhibit glucagon secretion. But one of the things that we found in an earlier study that was published in Diabetes in 2025 was that a lot of the effects of leucine are due to its ability to reduce cyclic AMP, which it does through both an intrinsic effect on the alpha cell and by stimulating neighboring beta and delta cells, which suppress the alpha cell indirectly. This left us with a model where alanine and arginine are excitatory to alpha cells. They promote glucagon release, and leucine is inhibitory to glucagon secretion.
And then there was a bit of a caveat with these findings, right? So, and that kind of led to addressing that caveat led to the next set of studies. Can you tell us about that?
What we wanted to do is try and understand if leucine was working through alpha cell metabolism in a very similar way to in beta cells, and working through mitochondrial PCK2 and pyruvate kinase. And so to manipulate those enzymes, we used 3 different tools. We used 2 alpha cell specific knockouts where we specifically deleted PCK2 in alpha cells. We also deleted 2 out of the 3 isoforms of pyruvate kinase specifically in alpha cells. or we activated pyruvate kinase with small molecule activators. So the first thing that we found was that leucine works in alpha cells metabolically in a similar way to beta cells, insofar as if you put leucine on an alpha cell, it can force the mitochondria to make PEP, and that PEP can work through pyruvate kinase to close KTP channels at the plasma membrane. We know that because the knockouts would block the effect on KTP closure. We also were initially excited because we saw that knocking out either one of those enzymes had an excitatory effect on calcium levels. And so we started to have a mechanistic basis, we thought, for how leucine could maybe suppress glucagon secretion, in part by suppressing calcium. But that turned out not to be the case at all. The effects of alanine and arginine to stimulate calcium influx in an alpha cell can be suppressed by leucine, but the effect on calcium doesn't matter for glucagon secretion. And that was a really big surprise. One of the most powerful experiments, I think, that our lead did was looking at calcium in response to combinations of amino acids. And when he stimulated calcium influx with alanine and arginine, he got a very profound increase in alpha cell calcium. And he could suppress that by activating the pyruvate kinase enzyme and suppress 90% of that. But that had absolutely no effect on insulin secretion. And so the conclusion we drew is that alpha cell calcium, even in the presence of low glucose alone, is plenty of calcium to fully support glucagon secretion from an alpha cell. All of that extra calcium that comes in response to alanine and arginine doesn't matter for glucagon secretion whatsoever. And we know that from all three models that we use, 2 alpha cell knockouts and 1 drug. None of that calcium matters for glucagon secretion. We think this is really important for the field to know, because if you read a textbook that explains the mechanism of how alanine and arginine stimulate glucagon, it's the textbook explanation is they work by depolarizing the alpha cell plasma membrane. Now let's unpack that for a second. So alanine, when it's transported, is co-transported with sodium, providing a way to depolarize the plasma membrane. And arginine is charged. So when it comes into the alpha cell, it's already a cation, and so it can depolarize all by itself. But a lot of this concept came from studies where people used wildly non-physiologic concentrations of these amino acids. So routinely 20 millimolar arginine is used. Arginine is only present in the blood at 250 micromolar. So if you think about that working through a membrane depolarization mechanism, it doesn't really make sense physiologically. And so why I think it's exciting, it's an exciting finding that the calcium doesn't matter for glucagon secretion, because it provides an opportunity for the field to go find out how alanine and arginine actually stimulate glucagon release. It's not the calcium, it's got to be some signaling pathway. It has to be either metabolized or perhaps working through a transceptor, which just means a transporter that signals. We don't know, but the field needs to go find this out, because the longstanding idea that they're simply depolarizing the alpha cell and triggering more glucagon because of calcium turned out to be wrong.
So Matt, I want to kind of circle back real quick to leucine, right? Because in addition to being an amino acid that is metabolized and important for building proteins, it also is a pretty good activator of mTOR. Speaking to my ignorance, how did you separate out mTOR signal, potential mTOR signaling, from what you've now actually found and reported?
We didn't look at mTOR signaling alone. I think the explanation for leucine suppressing glucagon release is due to its ability to lower cyclic AMP, both through a direct effect on the alpha cell and indirectly stimulating beta and delta cells, which are inhibitory. No one has really looked at mTOR signaling in a stimulus secretion coupling way at a very detailed level.
Okay.
The Bernal Mizrachi lab has done some nice studies where they looked at mTOR signaling on longer time scales, but we still don't know what's going on a second by second basis. In fact, that's a big hole in the islet field in general, is how other nutrient sensors like the mTOR for amino acids impact the alpha cell or the beta cell on a second by second basis. No one's looked.
Great. Thanks. Sorry, again, a quick question that popped up. So you've touched a bit on the models that you used. Maybe can you give us a little bit more rationale for the knockouts, and then talk about the in vivo phenotypes that you observed in those?
Well, in both the PK knockout, the pyruvate kinase knockout, and the PCK2 knockout, which deletes the alpha cell pep cycle, we found no impact on glucagon secretion ex vivo or in vivo.
Okay.
And that might be expected because remember, the phenotype of those knockouts is gain of function. There's more calcium in the alpha cells and more of a stimulatory effect. We're not taking away function. So it wasn't really surprising to us that the in vivo phenotype of those mice was normal. They release the regular amount of glucagon, and if we stimulate the alpha cells by giving insulin to a mouse, which causes hypoglycemia, the alpha cells are perfectly capable of responding to that.
But you did see a difference in the electrophysiology. Maybe can you comment on that for us?
So what we did find is that in both knockouts, and particularly the PCK2 alpha cell knockout, that blocked the ability of leucine to close KTP channels and influence membrane potential and calcium. But it's really difficult to say with certainty that the KTP channel is important for that effect that we see on membrane potential and calcium. The alpha cell KTP channels, unlike in a beta cell, they're 99% closed in alpha cells at the basal state. So it's really hard to see how the little additional bit of closure we get by activating the alpha cell pep cycle or the alpha cell pyruvate kinase could really impact the membrane potential and calcium influx in a profound way. So I don't think that that is the mechanism underlying the effects of these amino acids that we see. Again, I think that when the field moves forward to explain the effects of alanine and arginine, we need to look beyond what these membrane depolarization and calcium effects are. There's something else big going on, and we don't know what it is.
So, are there any other key takeaways from the work that you'd like to leave us with, or any other key questions that you're excited for you or the field to address in the future? And you've touched on this a little bit throughout, but take us home.
Well, I want to take everyone home by just a bit of practical advice. One amino acid that I didn't talk about today is glutamine. Every islet biologist who's doing an ex vivo experiment should have glutamine in their bath solution. It is an essential amino acid. It's not one that is sensed by the alpha cells, but it changes the phenotype of the other nutrients so profoundly that it should always be present. And so the effects of alanine and arginine on calcium, for example, require glutamine for the full efficacy. The effect of leucine, which is suppressive to alpha cells, requires glutamine absolutely. In fact, if we looked at the knockouts, in the absence of glutamine, there was no phenotype, which tells us just how important glutamine is for the alpha cell to function.
Great. Well, Matt, we want to thank you for joining us today and sharing your expertise, and also for discussing your recent work on amino acid regulation of glucagon secretion. I encourage the listeners to read the full article, which is in the March issue of Diabetes.
Today, we will be discussing a paper published in the March issue of Diabetes titled Activating Muscarinic Receptor Signaling in Intrapancreatic Neurons is Required for Parasympathetic Cholinergic Control of Pancreatic Cell Function. We are joined by first and corresponding author Noah Levi, who is an MD PhD student in the group of co-corresponding author Dr. Alejandro Caicedo in the Division of Endocrinology, Diabetes and Metabolism, Department of Medicine at the University of Miami Miller School of Medicine. Welcome, Noah and Alejandro. Thank you so much for sharing your work with us today.
Thank you for having us. We're really excited to be here.
Thank you very much.
So guys, we like to start with a question that we haven't prepped you for, a little icebreaker. And today, what I'm curious about is, and we'll start with Noah, if you were not in science, or I guess medicine, as you are pursuing a dual degree there, what career do you think you would have chosen?
I think something in the culinary arts.
All right.
Yeah, I like to cook. I like to joke. I'll give a long-winded answer. My brother is a musician and a piano tuner, and I'm in the field of science and medicine. My mom is an artist, and my dad is a liver transplant surgeon. So I joke that I got the science and he got the art. But I think in another, I think in another world, I could have gone down the artistic route, maybe through cooking, because I love to cook. I love food. So yeah, culinary arts.
I feel like that's not the first time we've heard that, which is kind of interesting.
Yeah. Too much time in the lab. You
Yeah.
know, protocols and recipes aren't that far apart.
Yeah.
Yeah.
All right, Alejandro, I guess I'll ask you the same question if you don't mind.
All right. No, I think, similar to Noah, my parents were both artists, painters, and maybe I decided to become a scientist because I was a rebel. So it's normally it's the opposite. An artist is a rebel, and your parents are more like the lawyers and doctors, but in this case, it was the opposite. I could have been a painter. I think, I don't know if successful or not, but I would have liked to do that. So when I started college in Germany, the choice was between going to the scientific part or to go to the arts academy. I don't know. So, but now I'm a scientist. I don't know if for good or bad.
Well, there's some beautiful immunohistochemistry in your paper, so
that's right. And
maybe that reflects your artistic.
And our listeners can't see it, but I do notice some art in the background there of your office. So.
Oh yeah.
All right. Well, I guess it's time to talk science now. So, Alejandro, we'd like to start by asking if there are any other authors or collaborators that you would like to acknowledge on the completion of this work.
Very important question. I think that there's a co-first author whose name is Alejandro Tamayo. He did a lot of the technical aspects of the slices as well as the perfusion studies. So he was instrumental in all of us to be able to conduct the work. Then there is Madina Sokolov, formerly known as Madina Mahmutova, who is also very important, who is now a senior postdoc in the lab, who is very important for this conceptual aspects of the work that we performed. And since this involved also a channel that was sort of not well known for us, which is the KCNQ2 channel, the KV 7.2 channel, that seems to be crucial for the transmission of the signal from the neurons to the target tissues. We teamed up with Dr. René Barro, who is a professor of physiology as well as of endocrinology within our university. So these are the co-authors.
Excellent. Science is definitely a team sport.
Yes,
absolutely.
All right. And then Alejandro, can you start by giving us an overview of your research program and then maybe going into a little bit on how that drove this particular project?
Oh, very nice. So we are interested in understanding the physiology of the pancreatic islet, and we sort of pioneered a lot of the studies related to the human islets. We were part of this effort to transplant human islets in patients with type 1 diabetes. And as part of the program here at the Diabetes Research Institute, we started working a lot on human islets. So, but of course, the human islets are embedded in the pancreas, and they don't do this work alone. They need to coordinate their activity with the body. And that's why we are also interested in how the nervous system affects the pancreatic islet function. So we did some work looking at the anatomy of the innervation of the pancreas in the human. And now we are using a mouse model because it allows us to interrogate different aspects of this innervation. So the islet, although the name says it's isolated, it's not an isolated organ. It has to be coordinated in its activity with the rest of the body. So that's sort of what we do. We're looking at the connections of the pancreatic islet with the gut. That's what Medina is doing in the lab too, and how the pancreatic islet communicates with the brain, not through the bloodstream, but also with nerves, which is the afferent sensory pathway of the vagus nerve.
Excellent.
So Alejandro, one last bit of background housekeeping here. We know that the endocrine pancreas is regulated by substrate, glucose, and in an endocrine manner, such as the incretins. And then the focus of this paper was its vagal innervation. Can you provide our listeners with a little bit more background on what was known about that vagal regulation of pancreatic islets? And then what are some of the gaps in knowledge that you wanted to address with this work?
Yeah, exactly. What is the novelty of the work, and what is it that distinguishes this work from what has been done in the past? We all know that if we stimulate the vagus, and it has been stimulated in many different species, you can see changes in glucose metabolism as well as changes in insulin secretion. Now, when you stimulate the vagus nerve, it goes to many different visceral organs. And that produces confounding effects because these organs can be contributing to glucose metabolism, and they indirectly can also change insulin secretion. So what is not known is what happens in the last segment of the parasympathetic innervation of the pancreas. So we know about the vagus, it produces some responses, but we don't know the contribution of the local neurons. The parasympathetic system is different from the sympathetic system in that it's activating some neurons that are located within the target tissues. And until now, we didn't know exactly what these local neurons are doing, but they are very important for the function of the parasympathetic system and how it controls the target tissues.
Very good. Very good.
All right. So, Noah, let's start getting into the nitty-gritty details of the paper. The first thing we wanted to talk about is you used some very nice mouse models to address the questions at hand. So can you tell us a little bit about the mice you used and the methods you employed to study them?
Absolutely. So I'll say, you briefly mentioned it. We had two main questions that we wanted to address in this study. The first was, how do local neurons in the pancreas respond to cholinergic input that's coming from the vagus nerve? And the second question was, how do these local neurons that live in the pancreas impact pancreas cell function? So we employed two main mouse models to answer these two questions. So the first question was to look at the neurons themselves. So to do that, we used a mouse model whereby we had a genetically encoded calcium indicator in cholinergic neurons, and that's the CAT GCAM3 model. We were able to basically visualize calcium activity in these neurons as a proxy for cellular activity. The second model was a DREAD model, and DREAD stands for Designer Receptor Exclusively Activated by a Designer Drug. So we are basically able to use this transgenic model to insert a molecule into the local neurons to be able to selectively stimulate these neurons in order to see what happens to the target tissue function. So I'd just like to add that the method that we employed for the totality of the experiments, aside from the immunohistochemistry, for all the functional experiments, we used the living pancreas slice model. So we're able to generate living pancreas slices that are 150 microns thick that maintain the architecture of the pancreas. You have ducts, you have islets, and most importantly, you have vessels and neurons that are still intact. So we're able to interrogate or stimulate the neurons themselves and interrogate what's happening in the target tissue.
Very nice.
So, no, I want to talk now about one of the primary findings from this article, and that was, I guess, in my opinion, the identification of intrapancreatic neurons as targets of and mediators, I guess, for vagal input. Can you walk us through the study that led you to this conclusion?
Absolutely. So this was primarily a study using immunohistochemistry with a few different markers. I would like to say that a lot of work had been done previously by Bertou to identify that these local neurons in the pancreas are in fact targets of the vagus nerve. But we used a method to show that the totality, so that was done with tracing. We used a method with a transgenic mouse model to observe the totality of intrapancreatic neurons as targets of the vagus nerve. So the method that we employed was with a transgenic model called FOX2B line 3 Cree, crossed with a fluorescent reporter mouse TD tomato. And this basically allows us to visualize all of the terminals, both efferent and afferent, sensory and motor terminals of the vagus nerve. We co-stained with a cholinergic marker called VACT, which stands for vesicular acetylcholine transferase, which is basically the enzyme that loads acetylcholine into the synaptic vesicles. So that's a cholinergic marker. And basically, wherever there was an overlap of the cholinergic marker with the TD tomato staining, we considered to be efferent terminals of the vagus nerve. And we found that those efferent terminals of the vagus nerve were exclusively going to intrapancreatic neurons, not acinar tissue, not islets, but just to these local neurons. This is important to us because everything that's coming from the vagus nerve has to go through these local neurons.
All right. Noah, you then uncover that these intrapancreatic pancreatic neurons respond to both nicotinic and muscarinic stimulus. However, only the muscarinic receptor signaling seemed to be necessary and sufficient to elicit responses from the islet or the pancreas. So can you detail those results and maybe how you differentiated those signals?
Absolutely. So I'll first start with the initial finding that nicotinic and muscarinic receptor signaling look very different in these intrapancreatic neurons. So when we stimulated with nicotine, which is a nicotinic acetylcholine receptor agonist, we see a very large calcium peak that's coordinated amongst the intrapancreatic neurons themselves. However, when we stimulate with a compound called oxytremerin M, which is a muscarinic receptor agonist, we get a response pattern that's very different. The response pattern is seemingly random, but very sporadic and long lasting. It almost looks like fireworks in the ganglia. When we moved on to the DREAD platform, what was really interesting to us that we didn't realize until we were maybe halfway through the study is that the DREAD model leverages the muscarinic acetylcholine receptor signaling pathway to generate responses in the target tissue. So previously, when we tried using nicotine to stimulate intrapancreatic neurons, we didn't see responses in the target tissue. However, we stimulated intrapancreatic neurons specifically. We were able to see, because of this muscarinic receptor signaling pathway, we were able to see responses in the target tissue that we weren't able to see with nicotinic signaling.
Very interesting. And then to kind of bring it back to Alejandro mentioned the potassium channels that you all study here. So you link it to potassium M currents as a novel molecular mechanism. Can you touch on those findings?
Yeah, absolutely. So we tried to understand why the muscarinic receptor signaling was leading to this firework pattern, long-lasting, reverberating response in the intrapancreatic neurons. So we kind of collaborated with Dr. Rene Barrow, and we were talking, and he suggested the M current as a potential mechanism. So the M current is really interesting. It's a potassium current that normally exists at resting membrane potential. And it's an outward potassium current that basically prevents neuronal hyperexcitability. It acts as a molecular brake. And it's called the M current, interestingly, because of previous studies showing that it's functionally linked to the muscarinic receptor, which is why it's called the M current, M for muscarinic.
Okay. Yeah.
So what's really interesting is that when you stimulate muscarinic receptors through the intracellular signaling mechanism, there's a concomitant closure of the M current. There's a concomitant closure of that channel, which basically leads to a level of depolarization that allows for neuronal excitability to occur, for action potentials to be generated more readily. So in the experiments, when I used different agonists of this channel to keep the channel held open while simultaneously stimulating the muscarinic receptor, keeping the channel held open basically led to a diminished neuronal response, which signaled to us that this channel, the M current, was involved in the neuronal response.
Excellent.
All right, Noah. You've done a great job of identifying vagal efferents, the intrapancreatic interneurons, the novel M current mechanism, but I'm a dumb physiologist. So can we go back to what I think about when I think about the pancreas, which is glucagon and insulin? What did you find from your secretion assays? Specifically, I think you did the parafusion assays.
Yes.
And how's this signaling affecting what we think about of the islets?
So what was really interesting is that we were able to find that specific stimulation of intrapancreatic neurons led to a modulation of insulin and glucagon secretion. Related to the insulin secretion, there seems to be some sort of cellular memory. So I'll back up for a second and say that we were only able to see changes in insulin secretion when we changed the glucose concentration to a permissive glucose condition.
Okay.
So even when we had neuronal stimulation that was not in the presence of a permissive glucose stimulus, we didn't see changes in insulin secretion. However, what was really interesting is that when we stimulated neurons prior to an increase in glucose concentration, we were able to see a significant increase in insulin secretion, meaning that there was some sort of maybe memory that the cells had of being stimulated because the stimulation occurred prior to the change in glucose concentration change.
And I think, if I remember right, you linked this back to the cephalic phase of insulin secretion. Is that correct?
Yes.
Yeah, I think that conceptually, the vagus is stimulated naturally when you start having sensory stimuli in your head, and they are transmitted to the visceral organs via the vagus. The idea is that during the cephalic phase, a lot of people think that it's already stimulating insulin secretion, but what this study is now showing is that it's more than stimulating directly the insulin secretion, it's actually priming it for an upcoming surge in glucose or nutrients in general. So it's preparing the cell to what is coming next.
Yeah, I love the idea of priming the pump here.
Yeah. And it is actually interesting. The next question I was going to ask is about the exocrine pancreas. You also saw an effect there. So can you tell us about those findings, and do you think that that is also a part of this process, right? It's interesting to think that the neurons might not just preparing to handle glucose from a disposal side of things, but also the digestion of nutrients.
Yeah, absolutely. They're both linked to the same physiologic phenomenon. But what's different is that whereas the control of the islet seems to be more modulatory, the control of the exocrine acinar tissue seems to be under more of a direct regulation of the intrapancreatic neurons. So if you look at the traces in the paper, there are many calcium transients of the intrapancreatic neurons in response to direct stimulation that are mimicked by the acinar tissue. They have a very similar number of events per minute, is how we calculated it in the paper. It seems like the intrapancreatic neurons are imposing their will onto the islet, where it seems to be more modulatory.
So, Alejandro, are there any other key takeaways from this work that you'd like to leave us with? Or maybe you'd like to tell us where you're going to take this work next?
Well, I think the most important takeaway here is that these local intrapancreatic neurons, they have their own logic. And in a way, this is very difficult to demonstrate because these postsynaptic neurons are embedded in the tissue. So, but I can give you an example in the heart. In the heart, the local neurons are the ones that are responsible for the frequency of the heartbeat and also for the strength of the heartbeat. So here we think that the pancreatic neurons not only relay the information from the vagus, but they add their own activity to shape the function of the pancreas. I think this is the most important takeaway. So we cannot underestimate these neurons anymore. And we have to make an effort to study them, not only in the pancreas, but also in other target organs of the vagus nerve. Now, where do we want to go from now on? I mean, first of all, we don't know if the neurons in the pancreas, they are specialized for the acidic tissue versus the endocrine tissue. Are there label lines that go to different cell types? We also have to explore the effects of these neurons on the vasculature, if they're able to modulate blood flow in the pancreas. Are they having an effect on immune cells, cells that are probably mediating the immune attack in the natural history of type 1 diabetes? These are projects that are undergoing in the lab. And of course, one thing that is very important is how does this apply to the human situation? So what are the conditions in the human pancreas? And I think, based on studies that we have done in the past, that it may be a little bit different in the human pancreas. We don't see so many axons within the endocrine compartment, but maybe what we see in the human is more a release of neuropeptides in the bloodstream that then will affect downstream endocrine cells. So these are things that we're doing in the lab right now.
Well, Noah and Alejandro, we want to thank you for joining us today and sharing your exciting data and expertise on vagal intrapancreatic neuron regulation of endocrine and exocrine secretion. I encourage the listeners to read the full article in the March issue of Diabetes. Thanks to you both.
No, thank you.
Thank you.
Our pleasure.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and today we are talking about the American Diabetes Association's scientific sessions. I believe this is the 86th year that they're holding the scientific sessions, and this meeting is taking place this summer, June 5th through 8th, in a place that's very close to my heart, New Orleans, Louisiana. Every year, this meeting brings together basic scientists, clinicians, industry partners, trainees, and really just about anyone who cares about diabetes research and care. We are excited to welcome back this year Dr. Mark Atkinson, who is serving as the chair of the Scientific Sessions Programming Committee, to discuss what we can expect at this year's meeting. Mark, it's great to have you back. Welcome.
And thanks, Kevin, for having me back. It's exciting to not only meet with you again, but to have a few moments here to share what happened in Chicago last year, but also what we see that will occur in New Orleans. It's going to be great.
Yeah, and congrats to you and the team for the scientific sessions last year in Chicago. It really was a fantastic venue, city, experience, the meeting, everything. It just went off, it seemed like, without a hitch. So that's a real credit to you and your team.
Yeah, well, I that's the key word that you said there is team. And I would be remiss if I sounded like I took too much credit here. It's a team effort. I'm just a cheerleader and tour guide, if you would. But yes, Chicago was a very special meeting. ADA takes attendees' opinions, and I'll have to say that that meeting was one of the most highly rated that has occurred in the last decade. So a lot of good things happened in Chicago, and our hope is to build on that, build on the symptoms, optimism, and progress as we move into New Orleans.
Mark, you've now led the programming for a few years, which gives you a rare behind-the-scenes view. Most of us show up to the meeting, we look at the app, we might circle a few sessions, and then we move on. What do attendees probably not appreciate about what it takes to build a meeting of this size?
Thanks, Kevin. So I'll just relay that this is my fourth time serving as a member of the planning committee, but again, my first time in terms of chair, and I began this journey some 30 years ago, and it has changed over the years. We've moved from submitting 3x5 cards with ideas to putting post-it notes on the wall, to the current situation of spreadsheets and having input from subcommittees. It's truly changed, but all for the good, in that the ADA Planning Committee really wants feedback from its individuals and constituents. We want to put forward a meeting that people want to attend. So it's not only an honor to serve as the committee chair, but I truly want to acknowledge the efforts of the sessions planning committee. It takes many, many, many hours to try and put forward a domestic and global effort that is meaningful to all.
And last year when we started, we chatted about your kind of history, your time with the society. I think last year was your 40th anniversary of attending the ADA scientific sessions. And this year, so you're breaking that 40-year mark and going into 41. I was curious, when you picture everyone arriving into New Orleans, their badges on, coffee in one hand, maybe a beignet in the other. They're walking into the convention center that first morning. What do you hope they feel? What's the energy that you're trying to project for the meeting for these attendees?
Well, you hit, Kevin, two of my favorite things here. Again, that chicory dosed coffee, as well as the beignets, and having all the little sugar all over yourself and having to excuse yourself wiping it off. That alone might be a reason enough to go to New Orleans. But
Absolutely.
This is an exciting time. You noted my years here, and again, I can just say there's never been a time in my career, and again, I'm old. I'm getting Medicare notices in the mail all the time, but yeah, but the sense of optimism and progress has never been higher. And again, building off of Chicago, where we had so many articles published around the time of the meeting for data that was presenting in the New England Journal of Medicine and other high impact journals showing dramatic, and I don't want to underemphasize that word or be seen as non-scientific, but we are clearly in dramatic times in terms of improving diabetes care. And the challenge was, in a way, and I'm being very transparent here, how do we take was great and make it better in terms of a meeting? I think there was a book a few years ago about going from good to great or whatever. That was a challenge for the committee. But I think we've come up with some ideas, and we are encouraged in that.
Yeah, what we touched on there is that it does seem like every year the science in this field is evolving so quickly. It's so rapid. It's really cool to look at this when you are researching care and treatment and therapies for patients, and watching these basic science discoveries move so translationally up into the clinic so rapidly. But it has to be quite a challenging task for the committee in trying to figure out what to elevate, what to expand on, where to create space for all of these new ideas. And you mentioned all these articles being published so rapidly at the time of the meeting. I was curious, as you and the committee are shaping the year's program, what are the specific areas that you intentionally wanted to emphasize, or how do you adjust compared to last year's meeting?
Right. Well, two things here. One is, and I'm probably the least best person to describe this, but I really feel like in diabetes, we're having a situation like Moore's Law, which has often been described to technology and advances there, in that there is a decrease in the time it takes to see a true innovation occur, and it is just building. And I feel like that's what's occurring in diabetes care, that things are just moving forward at such an accelerated pace that people need to catch up, and it can be hard. And that's what I think is an opportunity that the ADA scientific sessions affords individuals who attend. It is the ability to catch up, if you would, on what changes that have occurred. And it's not just or I should say that we as the committee, when you say trying to make things different over years, we as a committee, work very hard with ADA staff to try and make this a personalized effort, meaning not just one way, people speaking and then, hearing, but we're trying to create environs, if you would, where there's opportunities to interact and converse and make it a much more personalized experience.
Yeah, and we talked about how, intentionally building into the program these different experiences. When you step back and look across the full program, what are the major scientific currents that you think are running through it? I know that there are these exciting opening plenary lectures that the society always organizes and tries to get a deeper integration between potentially diabetes science and obesity. Are there other types of talks, or how do these talks integrate with the whole scientific program that you guys have developed? What stands out to you?
All right. So I don't want to put your audience to sleep here, but just to understand the complexity of that question. When the committee meets, there are actually 25 different areas of diabetes impact that are put forward in terms of needing representation. And those 25 areas fall into 8 thematic areas that we call educational tracks. And this is something that we've worked on with ADA leadership, where we're trying to say, if you attend 1, 2, or 3 of these tracks, that your educational experience would be dramatically improved and have a satisfaction around this. And these include areas from acute and chronic complications, behavioral medicine and nutrition and exercise, clinical therapeutics, the notion of epidemiology and genetics, a very important area in terms of immunology and beta cell replacement for type 1 diabetes, and then for type 2 and type 1 diabetes, insulin action, molecular metabolism, obesity and physiology, and certainly, last but not least, islet biology and insulin secretion. So essentially, on the one hand, you may think that attending the ADA may be like drinking from a fire hose, but I would rather put a positive notion on it, that you will, attendees at this meeting will have a smorgasbord of various different themes to choose from. So we really try and be inclusive in terms of providing the attendee essentially the A to Z's of diabetes.
And I love all of the emphasis across the board, from basic science to translational physiology, lifestyle interventions, therapeutic opportunities that could be pharmaceuticals or maybe surgical intervention. And I know at this meeting, it's always very exciting clinical trial data that's released and major descriptions of results of different interventions that are released at this meeting. Last year, it was there were several of these that were highly attended, standing room only, difficult for actual attendees to get into the rooms for the release of that information. So there's a lot of excitement around those types of talks as well. And I was curious if there's things that have been integrated around this year's program to accommodate for maybe more people in the room, or being able to disseminate that information more widely to the attendees at the meeting.
Yes, I can speak on behalf of the ADA that as much as I can say positive about Chicago, it did represent challenges to us in terms of room size. And as much as we tried to accommodate room size, it was just physically an impossibility to accommodate everybody. But despite those challenges, people were really positive, and we have taken extraordinary measures here in New Orleans to try and avert that situation. But again, at the end of the day, this was all for the positive, in that there were so many articles, as noted, that came out in very high impact journals and clinical trials. And you bring up a good point. The ADA has what they call innovative and late-breaking clinical trial presentations. This is part of the program. And the committee will look at these applications that come in typically 2 to 3 months later than the traditional suggestions for presentations. Highly competitive. But what they are is the representation of real-time, highly competitive, high-impact clinical trial results. And so these have been selected for this year. Just the last one, I think, was about 2 days ago. And they will really bring the attendee to, again, real-time and novel cutting-edge therapies for type 1 diabetes and type 2 diabetes, as well as obesity.
Yeah, it definitely is a challenge, I'm sure, from every year to try and predict, which rooms these types of talks need to go into and how to accommodate and facilitate the attendees getting in and out of those rooms. So it definitely a challenge, and it's something that has to be adjusted to on a yearly basis, I'm sure, but dependent, like you said, upon city amenities that are available in each conference center. And then another thing, we always try to emphasize here on the podcast is the trainee experience, for students, postdocs, even junior faculty who are coming to New Orleans. Maybe it's their first time to New Orleans, maybe their first time to the scientific sessions meeting. What would you encourage them to focus on at the meeting?
So I would first encourage them to go do, as you said earlier, go get some beignets and some coffee. That is to just have the experience there, and the jambalaya. But the the New Orleans, I think, the ADA has its rotations from its cities that include, Orlando and San Francisco and Chicago, as in last year, and San Diego. But I think New Orleans is kind of interesting. It introduces people to the Old South. It's a very vibrant city with an amazing history. And it's, again, not sounding controversial here, but when we talk about diabetes, but the food is amazing. So if you're into culinary arts or anything on that, you will have quite the experience there. But for young investigators, to get to your point, again, I think that the greatest opportunity is to, in one setting, see the spectrum of diabetes from A to Z. As in, there's so many meetings that are specialized these days where they're very focused. I'll call it siloed, and that may not be the best term, but they'll be very focused. And young investigators are often encouraged, for good reasons, to silo their research efforts in order to obtain funding or move forward in the clinical practice or become experts in one area. But I think what the ADA affords is an opportunity to not only foster an individual's interest, personal interest, but also to expose them to the other areas, make new relationships, find people that are not like you, but maybe have a way to make what you do better, and share thoughts about how to move forward. So I think the ADA meeting is great in terms of that, affording an opportunity for young investigators to expose themselves to the diversity and the A to Z's of diabetes.
I think it's really great. Mark, I always steal from our co-host who's not able to join today, Kirk, who says that the ADA is a place where you can find your tribe. It's a place where you can find people that are working in a subject matter or research topic that is very similar to what you're working on. They can turn out to be colleagues. They can ultimately turn out to be mentors, champions of yourself throughout your career. And as you are a member of this society over the years, they can encourage and help you grow and develop. And I think, Mark, you're a perfect example of this, being a member of the American Diabetes Association, the scientific sessions, attending those meetings for several decades. And so I think it is a really important point to those trainees and junior scientists as they're coming up in the field is the ADA is welcoming you to find your tribe and to find your group of people that you can be developing and working with throughout your career. And every year there are a few moments that people are attending these meetings and they come out of a talk and it just sparks conversations in the hallway. It's something that they take back with them when they're discussing with their friends and colleagues at their own universities or institutions. What did you learn at this meeting? What is the exciting thing that's going on in diabetes care and research? And I was wondering if, without giving too much away, Mark, what would you say about what's going to be happening at this scientific sessions?
All right, I'm going to answer that in a two-part response. Number one, I'm going to say that notion of find your tribe is absolutely brilliant and agree with it fully. The ADA offers the chance to not only develop new friendships, but collaborations. And in my particular case, again, with more than 4 decades, again, many of the relationships that I developed occurred through attending ADA. And so for young investigators, again, this is an extraordinary opportunity to meet people not only of your own age and peer group, which is clearly important, but again, to interact, walk up to the podium, ask a senior investigator, and develop something. I mean, these could be, career impacting, the opportunities there. But in terms of what is new or what we're excited about, or I should say I'm excited about, and the committee is excited about this year. One is the infamous elephant in the room, GLP receptor agonist in 2026. You don't go a day without hearing some news about this, and whether it's new drugs and then more than 150 of them actually in development, and how we're going to handle GLP receptor agonism in all of its forms. That'll continue to be a theme. And flipping on the opposite side, stem cell therapies for type 1 diabetes and potentially for type 2 diabetes. Very positive news has been reported over the last year in terms of this. And we at the ADA committee have dedicated multiple slots to try and hear what's the newest and greatest on stem cell therapy. Another notion that's a little bit old but continues to have life is the notion of the microbiome. And in particular, what is the role for the microbiome in terms of type 2 diabetes and obesity? Another personal favorite of mine, and one that, again, formed the basis for one of these late-breaking clinical trials, was a notion of seeing amylin as a novel therapeutic for diabetes and obesity. I predict that this would be, as you noted, Kevin, earlier, a room that will be packed out. We'll also
Absolutely.
Yeah, we'll also hear updates on the standards of care and definitions of overweight and obesity. And here, again, a lot of controversy erupted from a Lancet Diabetes and Endocrinology report last year about how to define obesity, role of BMI, waist circumference. It really controversial. And in an effort to address that controversy, the ADA is going to excitingly have a number of talks on that. And then you go into the notion of debates. And debates make some people uncomfortable. Not me. I love debates if performed properly. And we work with our speakers to make sure that debates are respectful and not targeting the speakers, but present both sides of an issue. And these, over the years, that we have introduced these, have been wildly popular. I mean, amongst the highest rated efforts. So to hear the two sides of the coin, if you would, Absolutely. I'm looking forward to that. Just a few other things. Looking at the impact of AI, artificial intelligence moving forward, and how that's going to impact diabetes care, nutrition. Wow, we turned the table, the nutrition pyramid around in the last year, and not getting political, but what is the impact of that for people with diabetes? That'll be an amazing topic for discussion. The changing demographics of diabetes, both domestically and in the U.S., is big. And then also some holdovers from last year about ketone monitoring, and then the availability now of continuous glucose monitoring. You can walk into a local drugstore now and get CGM, but to what benefit? I could go on and on here, but all you're hearing from me is excitement about the topics, and I'm sorry if it appears eclectic.
No, I think it's great. I think it really highlights the scope, the scale of the scientific sessions, and really goes back to if you are really interested in the research at the basic science level, at the clinical level, the clinical care and treatment of individuals with diabetes, I think this is the meeting you really need to attend this summer, coming up in June. And so I really thank you, Mark, for joining us and giving us a preview of what's ahead at the ADA Scientific Sessions. It is always nice to hear what's coming to the society, to the scientific sessions from the people that are helping shape the program. And to our listeners, there's still time to register for this meeting. So if you've been meaning to sign up, consider this your nudge. And just as important, the late-breaking abstract deadline is March 9th. So now is the time to get that exciting data submitted. To register and learn more about the program, head to professional.diabetes.org/scientific-sessions. And as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.